Hello, students, and welcome to my screencast on DNA for your test. There'll be three screencasts. There'll be one on DNA all the way up to mutations. There's a very short one on biotechnology. It's actually from last year's class. And then there's one on meiosis. So this will be the longest one, so let's rock and roll. All right, so DNA, there was a whole march to the discovery of it. We went over some of the scientific experiments. What's important in those experiments is that they worked in a claim evidence reasoning framework, right? And just like you do here at TASM, and we can look at them and see some good science and see figuring things out. So let's jump in. So these guys, Hunt and Sturdivant, figured out that there's genes and chromosomes and they're going to code for phenotypes or what we see. But the question was, is it the protein or the DNA that is the important material for heredity? You don't need to know anything about Hunt and Sturdivant, but you will need to know a little bit about this experiment. I'll give you information on it, and you just need to recall what happened and then get from that what can be the claim evidence reasoning. Okay, you put this rough strain into the mouse, and the mouse lives. This is an experiment conducted by Frederick Griffith trying to develop a vaccine for pneumonia. If you put the smooth strain in the mouse dies, and the smooth strain has this polysaccharide coat that lets it evade the immune system. Just like if you go out into a wadi and drink some water, if you heat it up first, you're less likely to get sick, because when you heat up or boil the water, it kills the stuff inside of it. So when he heated up the S strain, the smooth one, it didn't kill the mouse. But get this. If he heated up the S strain and put it with the original R strain, the R strain changed. It was transformed. It became deadly. It gained the properties of the smooth strain, and it then killed the mouse. So what's important from this experiment? What is the claim? Something transforms the bacteria. What's the evidence? Well, the R strain that did not originally kill the mouse now does in the presence of the smooth strain. What's the reasoning? They're not sure. He didn't know if it was DNA or proteins or what was causing this transformation. So hopefully you found that CER talk for the Frederick Griffith experiment helpful. Was jam on. This is the Avery, McLeod, and McCarthy experiment. This is lifted from BioNinja, a good website to help you out. Here, they're going to confirm that DNA is the transforming factor. So what they do is they took the heat-killed S cells. Those are the smooth ones that are bad, but they've been boiled, and they're going to add enzymes to destroy certain parts of the solution. So here, they take out all the proteins. They add proteinase, all right? It breaks down proteins. They then add R cells. Remember, those are the ones that are non-virulent or non-harmful, and the R cells become deadly. So if you look at this, the claim is that protein does not cause transformation. The evidence is that when you take out the protein, the R cells still become deadly. If protein was the transforming factor, then the R cells would not have become deadly because you took away the item that changed them. Same thing for RNA. You add RNase, ribonucleases. They chop up RNA. You added the R cells, and the R cells still became deadly. They were still transformed. So because they were transformed, it wasn't RNA that was doing it. It wasn't protein that was doing it. Something else was changing these R cells. This is pretty interesting. And this experiment's hard for students because you have to think a couple of layers in. You have to think in the negative here. You take those S cells that have been heated up and killed, just like the wadi water. You add DNase, which chops up DNA. And here, the R cells do not become deadly. They are not transformed, students. So if they're not transformed, it's telling you that in these beakers, DNA was doing the transformation. So therefore, transformation requires DNA, and DNA is the genetic material of the cell. So that's the Avery experiment. But like all good experiments, we find other ways to confirm it. And so there was the Hershey and Chase experiment with this blender. It's pretty cool. Some of you drew pictures of it in class. So what happens? Well, they know that sulfur is only found in proteins, right? In DNA has a sugar phosphate backbone and nitrogenous bases, but no sulfur in it. Sulfur is found in cysteine and methionine, some of the amino acids inside of proteins. So they attach a radioactive label to sulfur. They then grow bacteriophages in them. Bacteriophages are viruses that infect bacteria. So here they grow these viruses inside of this radioactive sulfur. They let them infect the bacteria, and then they shake them off in the blender. They go, take them off. They then centrifuge, which means to spin around quickly, the cells and the liquid, and they found that all the radioactivity was inside the liquid. This means none of the radioactive sulfur was injected into the cell. Well, viruses, students, are what we call obligate intracellular parasites. That means they're obligated or they have to use cells. Intracellular means within the cell, and they're parasites, so they hurt them. So they infect cells to reproduce themselves. So whatever the bacteriophage injected into the cell did not contain sulfur, so it was not proteins. Well, here, they're going to grow them in radioactive phosphorus, and in this example, we'll pretend it's blue. All right, the bacteriophages or the viruses now have this blue radioactive phosphorus in it. They infect the bacteria, and they inject their DNA in there. How do we know that their DNA was injected in? When we shake off the viruses, and then when we spin around the test tubes with the bacteria and the liquid in it, we find that at the bottom, where it's more dense, where the cell debris is, that is where the radioactive phosphorus is. In other words, students, the viruses infected the bacteria and injected their radioactive phosphorus in there. And since they're injecting the items needed to make more viruses, we know that they're injecting DNA, and we know that DNA is the genetic material for heredity. Pretty cool experiment. All right, around this time, scientist Erwin Chargaff figured out that the ratio of A's and T's and G's and C's are about the same for within a species, right? But it can be different between species. So this is going to give us our base pairing rules, where I told you A's and T's are equal and G's and C's. And then these scientists were working on figuring out the structure and function of DNA. And so we went through this in class, and we went through all their contributions. We know that Rosalind Franklin took this beautiful picture of DNA by looking at DNA in different humidities. And then Watson and Crick were privy to this information without her consent or knowledge, and they were able to piece it together once they got the information. They're able to figure out that DNA is a double helix, and they were able to put the nitrogenous bases on the inside. So if you're interested, you take AP Bio, we read the actual paper, and we go through it. And here you can see that DNA is a ladder, right? And we now know that DNA is the hereditary material. So that was cracked by Watson and Crick, and with help by Rosalind Franklin. All right, so DNA is going to be in a nucleus. It's what makes up chromosomes. It's also going to be wrapped around proteins that kind of fit in there. All right, sections of DNA is going to be a gene, and that gene could code for something like hair, whether or not you got a widow's peak, or what your thumb's like and your eye color, etc. All right, it's made up of nucleotides, and that the nucleotides would be the monomers, or what come together to make long strands of DNA. So a nucleotide would be a sugar. This is the deoxyribose sugar, the phosphate's on the backbone, and the nitrogenous base. It could be either an A, T, G, or C. A and G's are called purines, and T's and C's are called pyrimidines. All right, a purine and a pyrimidine always have to match up. You can tell A's match to T's because they have 2 hydrogen bonds, and G's match to C's because they have 3 hydrogen bonds. The real trick in figuring it out for Watson and Crick was figuring out that they're anti-parallel in nature. Just like Salt and Caboose Highway, one side of the road runs one direction, the other side of the road runs the other direction. It's the same for DNA, and that gives you that double helix structure. DNA is also negatively charged, which will be important when we go over electrophoresis, and that's because of this phosphate group. All right, here it is, and here's just a couple different models of it. So the inside is held together by weaker hydrogen bonds, and that's interesting, right? It's going to let us open up DNA and access the genetic code. The backbone is held together by stronger covalent bonds between the sugars and the phosphates. All right, this is just reviewing for you that a section of DNA is a gene that's going to code for a trait. So ask yourself this: what is the role of DNA in heredity? All right, hopefully you got an answer like this. It makes up genes and it's capable of storing, copying, and passing on genetic information in cells. All right, here's our base pairing rules: the A's, the T's, G's, the C. I told you the little mnemonic ashtray garbage can. That's just what my high school teacher taught me in the late 90s. Nowadays, you probably teach something else. So what chemical parts are the same in every nucleotide? Check yourself. Hopefully you got the phosphate group and the sugar molecule. You don't need to memorize the word deoxyribose. All right, what parts can be different? The nitrogenous bases. So if you remember in class, I showed you the pretty cool TED Talk with how much of DNA is the same. It's just amazing, right? Your genes and your friend's genes are the same for coding for glycolysis and how your body breaks down sugars. You know, the differences that you see and that you celebrate are big, like your eye color, skin complexion, personalities, etc. But that's just a small section of DNA, so it's really interesting. All right. Okay, see if you can come up with the other side here with the base pairing rules. Hopefully you got something like this, students. So A goes to T, T to A to T, T to A, C to G, A to T. All right, so that's pretty easy. How about this one? Can you figure this out? All right, here's the answer. So you can pause the video and go back through it, right? And this is just, you got to deduce A's equal to T's, G's equal to C, and overall that they equal 100%. All right, so these endless combinations give us the different traits. Okay, let's move on to how is DNA copied. So you're constantly making new skin cells, students, new cells that line your esophagus, etc. So how can we make sure that every cell in the body has the exact same DNA? And so the way they do it is we figured out that DNA copies itself semi-conservatively. So let's see what that means. It means that one strand is old and one strand is new. And so this is often called the most beautiful experiment in biology done by Messelson and Stahl to prove the semi-conservative nature of DNA replication. So an enzyme is going to unzip it. So next time you unzip your jacket, think of that enzyme. And that enzyme is called helicase. It's going to unzip it, and DNA polymerase is going to add new nucleotides to the opposing sides. Where do you get those new nucleotides from? From the food you eat, right? So you broke it down and you broke down the DNA that was in that food, and now you're going to use it to make new DNA for you. What's pretty cool about this is these will be identical due to the base pairing rules. So once you unzip it, the A's have to pair with the T's and the G's have to pair with C's. There can be errors in that, and those errors could lead to cancer, etc. But in general, that's the way it works. The enzyme that adds the new nucleotides is called DNA polymerase. Okay. So when you take AP Bio, we'll go over something called leading and lagging strands. And that's because DNA can only be added onto in one direction, the 3' end. That's a little bit beyond our intro biology course, but if you're interested in it, it's a fascinating work done by Dr. Okazaki, who figured it out, and they named Okazaki fragments after him. All right, so how can we do this in the time that's needed during S phase in the cell cycle? And it's because it happens at hundreds of places on the DNA molecule until they all match up and they've made it a complete copy of the chromosomes. Here's an example. Many different spots that it's going on. All right, in bacteria, it just kind of starts in one spot and continues until the whole thing is copied. All right, check yourself. And here's the Messelson-Stahl experiment. I said we were going to go over it in class, and we just never had time, but it's beautiful. It's really cool. If you think about it, they grew DNA in heavy nitrogen and then let it copy itself in lighter nitrogen. And so from that, you can see if you grew everything in a heavy nitrogen, well, when you centrifuge it, all the bands would end up at nitrogen 15. If you then took all those bacteria and let them reproduce one time in light nitrogen, well, then they'd be half old, or half N15, and half new, half N14. So the average between N14 and N15 is N14.5. So that's where all the bands came out. And here's what's cool about it, right? So now we're going to let it copy for that second generation. And so it's going to open up, and you're going to have, well, some of them were that new N14 strand. So they'll match up with another N14. And some of them were that old N15 strand, and it'll match up with new N14, new nucleotides. So you'll get half of them being all N14 and half of them being 14.5. Right? You would never have 15 again, right? Because they keep copying and there's no more N15 around. And so it just gets progressively more and more N14. So this is a pretty fascinating experiment. Okay, this we did in class and you all crushed it, right? So going from DNA to RNA to protein. We call this the central dogma of biology. What we've learned is that these genes can code for multiple proteins. There's ways that cells can regulate it through what's called intron splicing and exon shuffling. And they can make lots of different proteins, and that's what's really being studied now, and it's called the Human Proteome Project. All right, so genes are sections of DNA, right? But DNA's got to remain in a nucleus. It can't get out through the nuclear pores. So how can we make proteins that reflect the code of the gene in the DNA? Well, we give a transcript, and that transcript is called messenger RNA. Right? And messenger RNA is going to match the information in the DNA. See if you can do it here. Do you remember? First of all, just remember that RNA is going to be single-stranded. It's going to have the sugar ribose instead of deoxyribose. And it's going to use the base uracil instead of thymine, a U instead of a T. And I told you in class that RNA loves you. And hopefully you could use it for a promposal sometime. All right, so here we go. Let's see if you can get, I'll come back to these. I thought I had that as a problem. But anyway, it'd be UAUCGC. Okay, so we've made, and the enzyme that makes this is called RNA polymerase. So it's going to transcribe that DNA gene. The mRNA transcript leaves the nucleus and goes to the cytoplasm. In the cytoplasm, it's going to match up with ribosomes to make proteins. If they're free-floating ribosomes, the proteins will stay within the cell. If they're ribosomes attached to the endoplasmic reticulum, these proteins will get processed, they'll get sent to the Golgi apparatus and sent out of the cell. So what were we talking about in the nucleus with RNA polymerase? We're talking about making messenger RNA. There are 3 types of RNA you need to know. This can go into the cytoplasm to be read and turned into a protein. What are some other types of RNA? Well, there's ribosomal RNA. This helps to make a ribosome. This and a bunch of proteins together make a ribosome in the cell. And then there's transfer RNA. It looks like the letter T. It's going to carry the amino acid on it. And it's made up of a whole bunch of bases. But what's cool about it is it has this loop at the bottom, and it's called an anticodon. And it's going to match up with the mRNA inside the ribosome. And because it has a specific code, it's going to have a specific amino acid. So when you see the mRNA CCG, well, it's going to match up with GGC, and that would code for the amino acid alanine. All right, so let's see how it works, okay? Here comes the, I think I got a good picture of it coming up. Here comes the mRNA into the ribosome. It's going to have a sequence of letters. Remember, that reflects the gene inside the nucleus. The correct tRNA will match with it. So all proteins start with AUG. This tRNA is going to match up with it, and that's going to be UAC. That is the anticodon. This is the codon. I'm going to give you a review sheet on this the day before your test. So the anticodon carries amino acids with it. The tRNA carries amino acids with it. And that first amino acid would be methionine. Right? And then it's just going to keep on matching up. And I taught you how to read the chart, and you should bring your yellow chart to your test that day. Here's another example of it. So here's AGU, here's UCA, that matches up, and it's going to have a certain amino acid with it. Here's CGU, here's GCA. So this would be called a codon. Remember that we read them in sets of 3 or triplets. And this would be called the anticodon with the amino acid on it. All right, and just here's a couple of examples of it and what they would code for. So see, take out that yellow chart and see that you can do it. So transcribing is taking RNA polymerase to make mRNA. It's going to hold the information of the gene. And in translation is taking that information from the gene in mRNA, taking it to a ribosome and turning it into a functional protein. And that protein can go do work in the cell. And when you look at somebody, you're looking mostly at proteins. All right, last but not least, mutations. So in mutations, I gave you the sentence, the big fat cat ate the rat. Here, cat eat big rat. If you change something, cat eat big hat, you can change the meaning of the sentence. Some mutations are more harmful than others. Some of them, you can still figure out what the original sentence said, and some of them completely change it around. And so a frame shift mutation, what we call an indel for an insertion or deletion, changes the whole thing. So instead of cat ate big rat, here if we insert the letter A at the beginning, it changes the whole sentence to ACA, T, TB, GR. And it's really hard to figure out what that sentence was saying. So these are called a frame shift because it shifts the whole reading frame for the mRNA off. Sometimes you can have just a substitution error. It'll just change one letter within the strand. So here it changed TAAC. This T changed to a C, and it might change one amino acid. Or, so here's some examples of a substitution. We're going to change ATG to ATA. Here we'll change it to ATC, and here we'll change the first one to GTG. So normal overcoding for tyrosine. Here, this is called a silent mutation. Because even though you went from ATG, here you go to ATA, a lot of times that third nucleotide in a codon can be variable. And here UAU still codes for tyrosine. So in other words, that mutation didn't affect it at all. It still codes for the same amino acid. But here, if you change ATG to ATC, it now becomes UAG, which is a stop codon. And what that does is it immediately cleaves or cuts the protein from the ribosome. So this would chop it off. Or here, if we change the first letter, that's oftentimes changes the amino acid. And this would change it to histidine, CAC, which codes for histidine. So silent means it doesn't have an effect. Nonsense is when it converts it to a stop. And missense results in a single substitution. And so you went over these in the lens of muscular dystrophy. You should go back to that packet and see now if you can explain the difference between Duchenne muscular dystrophy and Becker muscular dystrophy. You do not need to memorize these terms. Those will do you no good later in biology. But what will do you good is being able to model and see how the effect of the substitution affects the protein. All right, here's just some examples of mutations that can cause different diseases in humans. And so there's great hope in CRISPR that I showed you in class of being able to fix these point mutations. And now we're at the point, literally no pun intended, right, where we're able to change DNA bases within DNA. And so human trials are starting soon on that. All right, I'm exhausted. I hope that was helpful in the words of Mr. Bozeman or Mr. Anderson. And let's get ready for that test.